I'm Jennifer Sun. I have the honor of serving as one of the co-scientific directors for MTM Vision, along with my colleague and friend, Tom Gardner. And this next session is all about translating science to clinical care for diabetic retinal disease. Here are my financial disclosures. And the next 10 minutes I'll spend giving you a quick update of what we've been doing over the last year. What's the work ahead? And I'm going to start with the metaphors that Robert introduced, the compass, the magnifying glass, the tape measure, to talk about our direction. So in this particular session, we'll focus on the direction towards an unimproved fundamental understanding of diabetic retinal disease. And this is our phase 1 scientific roadmap work that's responding to 3 key barriers to progress. You can't solve a problem you haven't defined, you can't find cures for human disease without studying the human condition, and you can't judge success unless you know what to measure. Our focus areas I'll be talking about are specifically the first and third answers to these questions, our diabetic retinal disease staging system and severity scale update, and the novel endpoints identification and validation work. So what have we been doing? Well, progress by the numbers. Robert mentioned the papers that we've published as a whole over the course of multiple years. In this last year, we published 4 papers. We have one ready for submission that's getting teed up for that, and 2 that are drafted and that we hope to submit shortly. I'll highlight one, Dr. Brian Vanderbeek, who's our first MTM Vision Research to Prevent Blindness Physician Scientist Awardee, who's here today, presented his work last year, 20-year trends in prevalence and incidence of diabetic retinal disease. So we hope to move forward the field in part by helping us understand the scope of the problem. Our scientific leadership presented 22 times at national and international conferences this year, and again, I'll just highlight one of them. Robert, Adriana Plevniak, and I went to the Diabetic Retinal Disease Congressional Briefing and gave that to a group of congressional staffers and to the emerging scientists in the NAVR program in September. Robert and Adriana then had the opportunity to go visit congressional offices, which was helped by the fact that she is a former congressional staffer, and really lobby for vision research in diabetes. Our participation at MTM Vision events continues to be robust. We had over 100 participants at our June Endpoints workshop, and there are actually 196, I think, registrants for today's symposium. We have the 2 multi-center studies launching soon in collaboration with DRC Retina Network, and we'll talk about that in a moment, and 2 major national and international endpoints collaborations with the Collaborative Community on Ophthalmic Innovation and with the Bridge Study Group, and Dr. Malvina Eydelman and Fabio Baucher will be talking about those respectively a little bit later. So let's talk about the clinical studies, because these are really the flagship kind of infrastructures that we hope will allow us to move forward both the severity staging and the endpoints work. We need to collect more data. Our working groups clearly told us that there are massive gaps in evidence, and we need to address those gaps in standardized and rigorous ways. So with DRC Retina Network, we're standing up protocols AR and AS, a natural history study, and a study of eyes beginning treatment with anti-VEGF for diabetic macular edema. These studies will include both the standard endpoints we use for retinal characterization, dilated eye exam, visual acuity, ultra-wide field fundus photography and fluorescein angiography, OCT, OCTA, reading speed, and Humphrey visual fields. But we've also identified some new or relatively new functional endpoints that we're interested in, including the manifold quantitative contrast sensitivity platform, the objective field analyzer, and the portable Retinal ERG. We'll do the ERG both in the light-adapted and dark-adapted states. And the goals for these studies are really, ideally, to develop new primary endpoints that are acceptable for use in clinical trials for regulatory approval to help speed the availability of new therapies to patients. I will say, throw-offs, though, hopefully will be validation of biomarkers as surrogate or clinical endpoints that can still be used to enhance research and clinical care, even if they're not suitable for registration trials. We welcome the European colleagues in EVICR-Net. We're hoping to involve 7 of those sites, and I welcome Stella and Tunde, who are here, who are part of that group, as well as our other collaborators in there. So what have we been doing over the last year? Well, to be honest, we had hoped to start these studies this year. I was hoping we'd get here and tell you we'd enrolled our first patients. What I had not quite counted on were the logistic and legal complexities of 7 sponsor contracts, each of which exponentially adds, I think, to the burden of getting this all together. But we're very, very close. I'd like to thank Cindy Stocktail, who's our operations day-to-day manager at the JABE Coordinating Center for DRCR, and Claire Calhoun, who's here today, who's our project manager for these studies, and both of them have done a remarkable job in the heavy lifting of getting all of both the sponsor contracts kind of consistently and for near final execution, as well as working towards finalizing the protocols, working towards finalizing our electronic case report forms. These studies have been get reviewed by the IRB already and are approved to start whenever we get the legal stuff sorted out, and we're hoping to start certifying our first sites actually within the next couple of months. So I'd also like to thank our protocol chairs, Tom Gardner, Leanne Labriola, and Mohammed Al-Masri, who've helped to lead the scientific charge on the studies. We've not waited for these studies to start to begin some of this work. So our Joslin Diabetes Center team has brought in the 3 instruments that we're using in terms of function and has done some initial test timing and feasibility work. Kamran Ashrafzada and Milan Bahi are our study coordinators who have done a fabulous job working with the instruments and giving us very useful feedback that we hope to bring out to the DRCR sites to make streamlining and ease of the tests better. So for example, for the manifold contrast sensitivity platform, they've had a lot of experience with this test at this point. They've emphasized to me the importance of good test instructions, giving a brief overview so patients know to expect 3 letters on each line, that the letters will get harder to be read from left to right, and that there's a box that appears briefly that gives a sense as to the size of the letters. It's helpful to let the patients know it's normal not to see all the letters. In fact, we're looking for that intersection as to where the letters are no longer visible. And running the test, just knowing that there's ability to go to the back button to repeat a line and ensuring that the participant doesn't lean forward, because that changes the visual acuity measurement. The objective field analyzer, we've learned that a dark and quiet room is important for this test, in terms of eliminating distractions, having artificial tears available to reduce artifact from dry eyes, and just letting patients know that occasionally they might lose fixation, but we can restart the test if that happens. Just turning the device on a few minutes before we start the testing is also important to streamline the visit. In terms of the ERG, they've worked at trying to figure out how to best attach the arm to a table or to a chair to give extra flexibility for positioning the device. But they've started to use eye patches for the patients, which is more comfortable than having the patient hold one hand over their eye. Emphasizing the importance of prepping the skin to placing the skin electrode, and proper placement of the skin electrode is critical for getting a good signal. And then in the dark adaptation, we've learned from early experience that just even having a red light lamp there, that doesn't affect the dark adaptation, but lets the coordinator see just a little bit to be able to position the patient, and the cable attachment is important. One of the things we've done is look at the average test lengths. And so Kamran and Milan have taken DRCR visits from other protocols and tracked the length of time it takes us to do these procedures. These are going to be long study visits, and they've added on the testing times for the new devices as well. When you add all of these up, I will say we're looking at an average of 4 to 5 hour study visits. This is not easy, and so it's going to be really incumbent on us to figure out how to streamline and how to give our clinics the best advice in terms of making the procedures faster and easier. As we've done this, we've also moved ahead in parallel with starting to build the consensus as to how do we move down the path of validation for endpoints as we start to get the data in from these studies. In June, we held a workshop that convened experts across clinical care, research, industry, and regulatory institutions that addressed these 5 areas of clinical assessment of retinal structure, cellular and molecular markers, assessment of function, validating clinical outcomes, and translation to action. And Konstantina and Sampani and Ward Fickweiler were good enough to transcribe those sessions, and they should be being submitted for publication shortly. They're here as well today, working on writing up a report of this symposium. Some of the early lessons we learned from these discussions were the importance of including endpoints relevant to the entire retinoneurovascular unit and prioritizing early-stage biomarkers in our efforts to see are these validatable as primary endpoints, assessing both structural endpoints and functional endpoints, and then integrating multimodal structure-function metrics, utilizing cellular and molecular phenotyping, and in order to support regulatory qualification of endpoints, working through launching these multi-center prospective clinical studies, harmonizing the imaging and analysis protocols, and standardizing performance benchmarks for AI tools. So there's a lot of work that's been done. There's a huge amount of work in the road ahead. We will continue to forge alliances across our academic, industry, lay community, and regulatory groups to encourage incorporation of promising variables in our major studies and trials, to standardize variables across data sets, to promote sharing of data sets, and finally to build consensus across stakeholder groups to support the identification, validation, and adoption of promising endpoints. I want to thank everybody that's involved in this work, and I'm looking forward to the next series of talks. I'll introduce them here because I won't introduce the speakers in between talks, so I'll just have you come up and launch into your talk as the next speaker leaves. But there are a few key questions that we're hoping to address in this session as we talk about translation of science to clinical care. Number one, why do we need new endpoints and staging systems for DRD? Riecke, unfortunately, who was supposed to give the talk, had a family emergency, but Fabio Baucher is going to pinch hit for him, and he'll talk about what can we learn from negative studies. Secondly, how is MTM Vision collaborating with partners to build community consensus? Dr. Malvina Eydelman will talk about CCOI and the work that we're doing together. What is the path to validating a promising new endpoint? Lloyd Aiello will speak specifically about retinal nonperfusion. And then finally, Chris Derman, our lay advisory chair, will wrap it all up and talk about how do we ensure adoption of this work as we think ahead. So I'll welcome Fabio up to the podium. And again, want to just acknowledge all of you that have participated, everyone who will be participating, and thank you so much for your efforts.
Okay, so I'm stepping in for Kai. Actually, he's my dear colleague from Bayer. Last year, we did the study together, so that's why I'm here. So this is translating science to clinical care for diabetic retinal disease. This is the next is what can we learn from negative studies? Yeah, this is something what happens pretty much ahead. 80% of studies fail in the proof of concept, so the proof of concept stage. So this is something that we need assessment. We need to do. We know that, Panorama, talking about NPDR patients, Panorama was a great success. So it was a great success in terms of effect of efficacy on the main endpoint, which is recognized in the FDA territories, so two-step DRSS changes in the study eye. And as you can see also here in the sham arm, the two-step improvement is very low, but as we know, both the Q16 and the in the Q8 and the Q16 actually arm were very, very effective. And also the event rates in visual threatening complication were pretty much different and significantly different in terms of aflibercept versus sham. As you can see on the left, very much left-hand side, 60 to 40% in 1 year to 2 years. This is the event that no treatment is leading to for those patients. And we'll come to that in the DRCR Retinal Protocol W. As you can see here, the adding Panorama was DRSS 4753 patients. In Protocol W, we also had 17% of DRSS 43, and anyway, around 4 years, still around 60% of vision-threatening complications, which were significantly reduced by treatment with aflibercept. So what is the point here is that apparently after 4 years, vision loss is still there and is the same as the sham treatment. So treatment with anti-VEGF is not really providing any effect, not even at 4 years. So the agency, what is the agency feedback? The DRSS improvement across the regions, Europe and US, two-step is really an important measure. Three-step for systemic treatment. Vision-threatening complication, anyway, is inconsistently considered and defined across territories. And PDR and CIDME are used as a composite or separately, depending on the regions. So what is important is that there is no agreement on the endpoints here, how to measure that. And BCVA is still the important. So the study that I'm going to present is a study with runcasigwat, proof of concept study with runcasigwat, an SGC activator, soluble guanylate cyclase, acting on the beta subunit on the heme group where actually the oxidation is the oxidation due to long glucose levels, long high glucose levels, has impaired the binding of nitric oxide to the soluble guanylate cyclase, impairing the production of cyclic GMP. Runcasigvat is agnostically linking and binding to this subunit, restoring the production of cGMP, and this was kind of showed in OCTA in healthy rats, and as well in streptozocin in type 1 diabetes-induced rats. So all the preclinical was there, the rationale for the study, the NEON-NPDR was there. NEON-NPDR was a randomized placebo-controlled mass trial with an oral compound, and this is why we have, as you can see in the upper line, the titration of the daily daily administration of runcasigvat, 30, 60, 90 milligram on a weekly basis, and then for 1 year with 120 milligram. This dose was the highest dose that was tolerable. So not about efficacy, it's about tolerability for systemic compounds. And it was, let's say, compared to placebo, DRSS 4753, good vision patient without CI-DME. And the efficacy endpoint was a two-step DRSS improvement, plus some, as you can see in the efficacy endpoint, change in retinal perfusion markers, which was a sub-study. The main efficacy outcomes were presented this year at ARVO. As you can see, the 0 is the leading column here. Big delusion for us, for the Bayer team. There is no effect. And just a few patients had one-step improvement in DRSS, and just one patient in the placebo arm. So pretty disappointing results. And vision-threatening complication, no difference. There is a slight separation, as you can see, around the middle of the curve, which is the 6 months time point. But then the curve, they regress to the mean, and they are absolutely, there is no difference. They're superimposing. So definitely a flat study, positive preclinical data, a lot of thoughts about how this mechanism of action could improve perfusion in tissues of retinal disease patients. Now, but looking at the others, at the other endpoint, so, and this data are not presented yet, will be presented very soon in a paper. So you can see that visual acuity, patients were starting above 79 letters. And as you can see here, there is between runcasigvat and placebo, and we are talking about more than 100 patients. So pretty decent sample size. There is not much of a difference, but also the variation, the standard error, which you can see between brackets, is good, is low. So it means that this is a reliable measure. The same is happening for leakage and for central subfield thickness or intraocular pressure. So this main activities, these main measure endpoints that were supposedly be influenced by better reperfusion actually didn't change much. But let's look at the sub-study, at the OCTA sub-study, looking at the foveal avascular zone on the area. As you can see, there is also not really difference, 328 versus 680. But you can notice between brackets the big standard error, which is accounting for a high baseline variability. And you can see also with the perfusion density or vessel length density analyzed with a skeletonized method, you can see also that there is no difference between runcasigvat and placebo. Again, anyway, quite a bit of a standard error in that respect. So what did we learn from endpoints in NPDR? I have to say that we are proud that because the study was well conducted, the sites were responding well to the treatment, to the oral treatment, giving the option and recruiting patients for a good, let's say, with a hope that a good treatment and sustainable treatment, oral treatment could be there. So coming from the regulatory authorities, efficacy endpoint, there is no consensus between regions across how to interpret DRSS changes. It is acceptable in the European and Asian territories to use DRSS as a primary endpoint, but it is absolutely not enough for approval. There is universal acceptance for vision in BCVA, for vision as BCVA as an endpoint. In terms of proof of concept, here things are getting more interesting, because here we're going into what can be used for novel endpoints. And obviously, BCVA or any measure that we have seen was not sufficient to see a change in baseline. But the assessment of retinal non-perfusion, let me just concentrate and focus on the assessment of retinal non-perfusion. It is still not a useful endpoint, at least what we have shown, because the variable baseline in assessing foveal avascular zone and leakage actually is still a bit too imprecise. Now, where this is coming from, what we have seen is that, well, is patient dependent, because sometimes the patients are really moving a bit, and the quality of the images are not that, let's say, sometimes are a bit not sufficient. So I would like to thank the steering committee, Professor Shubhaseva Prasad and Professor Bressler at chairs, and a lot of other people contributing to the study. Thank you so much for your attention, and stay for the questions.
I'm very, very excited to tell you about an incredible partnership that we have created with Collaborative Community on Ophthalmic Innovation and MTM. For those of you who don't know what CCOI stands for, it's Collaborative Community on Ophthalmic Innovation, and it's a very unique platform that aligns all global stakeholders, patients, this is why we're all here, researchers, manufacturers, investors, clinicians, regulators, payers, advocacy. Everyone is invited to be at the same table so that together we can create a novel engine for ophthalmic innovation. Our mission is to remove the barriers between groundbreaking ophthalmic medical products and patients who need them around the globe, and to unlock the eye's full potential in innovative medical products to advance both ophthalmology and the broader healthcare. Our structure is getting quite large. We have 17 work streams, and each work stream has been doing incredible work. But when it comes to inspiration, there's truly no work group that can compare with MTM. I feel incredibly privileged to have the honor to work with this team who day after day bring passion and dedication that is truly contagious. Together, we have created a DRD Cure Accelerator. It's a moonshot for eliminating vision loss from diabetic retinal disease. It's with that concept of breaking boundaries and going where no man has gone before that we have put together 2 first-of-a-kind sessions that we did last week. We all heard that there's no question in anybody who is in this room that we need to develop novel endpoints. But, doing the same thing the same way and expecting different results, we all know that's not going very well. So what did we do? We tried to figure out how can we use AI to change the paradigm, to change how long it takes to have consensus for endpoints for DRD, both for clinicians as well as patients. So on 10/27, we held an AI-driven insights session to advance consensus on DRD endpoints to transform medical product developments. And on 10/30, we heard from patients directly in order to drive faster cures in diabetic eye disease. The first question we asked from the professionals was, what are the limitations of our current classification scales for eyes at risk for vision loss from DRD? 3 specific themes emerged. Updating classification scales needs to happen now. Participants underscored the limitations in existing scales to accurately stratify disease risk, calling for updated criteria to incorporate systemic and functional health indicators. Impact of systemic health. Discussions revealed that interconnectedness of systemic health issues with retinal disease progression, indicating a need for integrated care approaches. And challenges in retinal scanning and AI integration is something we kept hearing over and over, emphasizing the reliability of AI technologies in improving diagnostic accuracy. Next question we focused on was what key ophthalmological tests should be performed in people with diabetes for both patient care and research purposes. The three themes that seem to be overwhelmingly agreed upon is that ophthalmological testing and procedures need to be refocused. Participants stressed the need for comprehensive testing, including OCT, fundus examination, and VA to identify early signs of retinopathy. Technology in ophthalmology was another very hot topic. The potential of AI and telemedicine came up again and again as the way to improve access and diagnostic accuracy was a recurring theme. Patient education and communication, which is a theme that we kept hearing this whole morning, was again reiterated here. Should testing change from early to late stage DRD? And if so, what would you recommend? We asked our participants. All of the participants agreed that there is an early and late stage diagnostic testing that should be done a bit differently, that education and awareness in eye health is of paramount importance, and they all talked about the role of advanced imaging techniques, and OCT kept coming up over and over, as well as fundus photography, as recommended valuable tools for early detection. So overall, from our professional session, we heard that we need to expand accessibility of imaging technologies, enhance patient education and engagement, and integrate AI and data. Then we took the next step, which is hearing directly from patients. We tried to listen to individual stories for how does your vision loss or fear of it affect your daily life and how do you cope? Participants highlighted that this is significant disruptions in day-to-day activities, stressing the challenge of adopting routines to accommodate diminishing vision. We also heard that managing diabetes is critical in mitigating vision loss, emphasizing the necessity for control over blood sugar. Emotional and psychological impact of the disease, as expected, was a very big discussion point. Next, we asked, what do you want your healthcare providers to know about how vision loss or fear of it impacts you? Empathy in patient interaction was something that we heard and was reiterated, as well as a need for consistency in vision care follow-ups and education on preventative care for vision loss. Last but not least, we asked, what worries you most about how diabetes-related vision loss might affect your life in the future? The patients talked about the impact perceived quality of life. They underscored the value of shared experiences and talk about the financial challenges of medical care being a major concern. So in short, the key recommendations from the patient session was the independence versus dependency, communication gaps in healthcare, community support was essential, and that financial strain is significant. What we learned is that whilst the AI-enabled tools do accelerate the analysis, and it's something that would take months to distill, only took hours, we do have some bells and whistles to figure out in order to fine-tune this process. And with that in mind, I truly believe that MTM took their commitment to being the moonshot for eliminating vision loss with these sessions, utilizing AI, to its true mission. Together, I look forward to continue to collaborate, innovate, and advocate so that together we can end vision loss from diabetes and see a future full of possibilities. For those of you who can join us, I invite all of you to take your phones and take a picture of the QR code so we can continue this conversation together in Hong Kong to collaborate, innovate, and illuminate. Thank you very much.
Good morning, everyone. We're going to move here a little bit into where I'm going to show you some data and so forth, but mostly as an example of how, looking at some of these endpoints where more data is coming, what are the roadblocks along the way, and how do we move these forward, and where things can be different, but at the same time, where we try to make progress with what we have. So it's building an evidence case and consensus around retinal nonperfusion and diabetic retinopathy. I'm not going to complete this today. That's not the point. But rather look at the role, evaluation, and a roadmap towards an approval endpoint here, looking at a specific area that has quite a bit of data. These are my personal disclosures, and they're mostly not relevant other than the fact that I have worked with DRCR in the past and present, and that what I'm saying today is not a direct representation of DRCR. I'm not speaking for them. Now, take a look at this. I think many of you know what this is, and there was actually a time before the DRSS. Now, many of us have a hard time remembering it, and some of you out there probably weren't born then. But the fact that we can say that tells us that this has served us very well for a while. This has been well associated with progression of disease, well associated with visual loss, has supported where we've gone to date. It is clearly not all that we need now, and in some day it may not be present at all. But the point is we have to take this and build from that. And indeed, all the initial data suggests that we may have to be adding to it rather than replacing it initially. Now I'm going to talk about retinal nonperfusion as you look here from chronic hyperglycemia through the neuro and microvascular damage. I had a neuro in there, Tom, just so to make sure we remember that. All the way down through the retinal ischemia and vision-threatening complications. Retinal nonperfusion in this morphological and pathological change is dead center. And as you can see across the bottom, some of which I'll go through as we go through today, it is associated with many things, including increasing disease severity, changes on DRSS, vision-threatening complications, need for treatment, and to some degree in the later stages, actually best-corrected visual acuity. So I'll spend a bit of time talking about RNP as an independent factor for disease progression, but also point out that it has been shown to have a direct impact on local visual function as well. All right, so how do we get across this winding road to a regulatory approval of RNP or something similar? And again, it is a winding, twisty road, and when you look at that, there are a wide variety of obstacles along the way or decision points that we have to think about, because every single one of these can be a big stop, as you heard earlier. So the question is, how do we look at some of these various things to allow us to go to the next step? One is the scientific rationale. Does RNP or whatever endpoint you're looking at need have a relevant scientific rationale for the pathology? And is it related to the onset and worsening of DRD? That then opens up to look at test parameters. How do you look at these various aspects? Because one needs to remember there are different ways to look at the same endpoint, and there are different places you can look at the same endpoint at different times, and all those can have an effect. That then opens the discussion of clinical relevance. Does evaluating something like RNP allow us to identify those at risk of vision loss? And what does the assessment of this add to DRSS? Is it any better than what we're using now? That then opens up the clinical relevance, one of the key points that gets very difficult near the end, showing do the interventions that we're looking for affect this, and does this then improve visual outcomes that you need, whether that be progression of disease, complication, or visual acuity. And finally, building a community consensus. You've heard about that, and you'll hear a good deal about this in the talk after mine as well. So I'm going to start with some of the data from Protocol AA. As you all know, this was a DRCR study of 544 eyes that were evaluated annually. Each patient had at least one eye with NPDR and without center-involved DME, and there were annual visits for 4 years. Now, I'll show some of the outcomes that are not non-perfusion, but are relevant, that were shown to be associated with various important aspects in AA. First of all, eyes on wide field fluorescein that had lesions predominantly in the periphery had a 1.4-fold increased risk of disease worsening over 4 years. This was consistently present within all DR severity subgroups, and DR worsening was associated with the worst lesion grade on ultra-wide field images for HMA, IRMA, and NVE. As I'm going to show you here, what's also interesting is the modality, even in the same area, made a difference. So here we're comparing the 7 standard field, the masked area of the wide field angiogram, with the exact same area, the masked, of the ultra-wide field image, and then asking is, if the lesions are in the FA more than in the masked color, is that worse? And indeed, in terms of disease progression at 4 years, it is, with the HMA progressing 47% if those were present compared to less, and then statistically significant. Same for IRMA, and same for the onset of NVE, but not for venous beating. So among eyes with NPDR using ultra-wide field FA, there were markers for increased risk of disease worsening that were not detectable using standard ETDRS field photography or ultra-wide field color imaging alone. Now, disease worsening was associated with these lesions, as I mentioned, and the more severe the lesions were, particularly in the periphery, we had a 2-fold increase in disease progression if you had NVE more severe. If the IRMA was more severe, it was 68% increased progression, and if you had HMA more severe, there was a 90% increased risk of progression. Now, of course, the interesting about these is these can all be related to underlying mechanism of diabetic retinopathy at whatever level you want to look at, but including down towards the common pathway for the late-stage vision-threatening complications, which is VEGF, which can cause these lesions, which is associated with these lesions, and of course is associated with non-perfusion, particularly if this non-perfusion induces retinal ischemia. So please remember that retinal ischemia and non-perfusion are not, they're closely related often, but not the same. So retinal non-perfusion can be found in eyes without clinically visible or with very mild disease, and it generally increases with disease severity. It is associated with an increased risk of DR worsening, and it's associated with a variety of biomarkers, including retinal leakage, central macular thickening, predominant peripheral lesions, as well as intraocular VEGF concentration. Now, here I'm going to be talking about the non-perfused areas from ultra-wide field fluorescein angiogram. In the blue, that's non-perfused area, the total area that was measured. Those are the bars that you see there in blue. And then a measure that has been normalized, that is the amount of non-perfused area compared to the total area of the retina that was imaged in that photograph, because obviously that can change. So this in many ways, a normalized index is perhaps more important, but as you can see, they correlate very well. And as you can also see, as disease severity gets worse, according to DRSS, there's a very significant increased association with more non-perfusion and a higher non-perfusion index. Now, what about the areas of the retina? Well, if you divide up the areas this way, it's roughly 1/3 of the retina as far as periphery, 1/3 mid-periphery, and 1/3 posterior pole. And does this make a difference? And indeed here, again, if you look at the total non-perfusion by these areas, again, going from no disease on the left all the way to more severe diseases on the right, as the NPI increases, you see that there are changes, particularly in the mid-periphery and the posterior retina as the disease worsens. And indeed, the mid-periphery shows a 1.5-fold increase in non-perfusion as the disease severity goes up, and the posterior retina shows a 6-fold increase as it goes up. Now, please note here, we're looking at the entire retinal areas, either the total or each of these, not just the macula, central macula, as was being talked about in the presentation before. So in eyes with NPDR, worse DR severity was consistently associated with increasing non-perfusion ultra-wide field, and it was initially located mid-peripheral across all of the severity and progressed posteriorly. Here's the data for the DRCR across the whole thing, and in those with the lowest NP, or no non-perfusion at all, that's the bottom curve, and with the highest section of NPI on the top, you can clearly associate with progression over time. That's a 77% change in risk at the end. Now, more importantly, if you look at individual changes in NPI, as what the effect of that is, so this is now for every 0.1 unit increase in NPI, you can see in the posterior pole, that's associated with a 35% increase in disease worsening. And if you look at the disease type parameters that are a problem, like development of PDR, that for every 0.1 increase in NPI goes up 16%. And if you look at development of vitreous hemorrhage, that's going up 30% for each 0.1 increase in NPI. But you may very well, what's also important is this confounded by a variety of aspects. And here, this is adjusted for DRSS level, gradable area, ultra-wide field area, correlation between the two study eyes, and risk factors for each patient, the age, diabetes type, duration, A1C, and albuminuria. And you still see it significant with a 14% increase with every 0.1 unit increase. So because of timing, I'm just going to state that what we've also seen is that there's a very tight association of the complications of PDR, CIDME, and PDR with the retinal non-perfusion, particularly in the early stages, where this is the level of non-perfusion in those that had no progression. But here, whether it's PDR, CIDME, PDR, PDR, and CME, those levels are increased. And this is particularly true if you look at PDR and CIDME or PDR, so driven primarily to PDR. So I'm sorry about the timing here. This is also broken down by posterior pole, and we see the same thing here. Early onset PDR tightly associated with increased non-perfusion posterior pole or mid-periphery. So these are the increases that you will see. So in summary, retinal non-perfusion is tightly associated with various of the key parameters. I went through very quickly on the others, but there's very good evidence to show that it is very predictive of early onset PDR, particularly within a year or 2 years, so very significant in that standpoint. So when we go from looking at the various potential problems, we see that there is good scientific rationale already. We have test parameters that can give us this association. Is it the best? We never know. As what we know so far, there are probably better, but so far it's associated. There's good clinical relevance for this. Is there a clinical relevance in terms of interventions? This we do not know, and we need that, of course, to define these further to build community consensus. So with that, I'd like to thank the folks that were involved in this, including Bergen Engelheim and Marianne and Hannah particularly, who helped do the secondary analysis that I had to run through, but were very important. So thank you.
Good morning, everyone, and thank you for having me today. In the interest of time, I'm going to try to move quickly through this. But before I start, I'd like to give a little bit of background about me and why it's relevant to today's conversation that I'm going to have with you. I'm a type 1 diabetic of coming up on 37 years now, and I don't have diabetic retinal disease per se, but I do have diabetes-related visual issues. I've had high intraocular pressures for the last 8, 9 years now. And so I have essentially the onset or potential onset of glaucoma. I was trained as a scientist. I have a PhD in neuroscience. I was a professor at the University of Utah for a while, studying substances of abuse. And now I work for Myriad Genetics as a senior scientist, and I work to design, oversee, and execute the verification, validation studies that are needed for regulatory approval of the genetic tests, both the analytical studies and to some degree the clinical studies. And so I guess I have a somewhat unique perspective as both scientist and patient, but I stand before you today as a patient and as an advocate, and what I'm going to ask of you is effectively my title. Please engage patients to accelerate the clinical adoptions of all the advances that you're working on right now. Do what you can. I could end my talk there. Maybe we should just save the time and move on to the panel discussion. But I'll give it a little bit more color. Why do I think that we should engage patients? Well, there are many, and I'm sure you can think of many, highly effective therapies that have minimal or far lower than expected clinical adoption than they should. And we'll talk about today a case study that affects me personally, and that is the uptake of continuous glucose monitor use versus insulin pumps in type 1 diabetic adults as well as children. Many of you are familiar with type 1 diabetes, but in case you're not, there are some relevant things to discuss. Type 1 diabetes is an autoimmune disease. It destroys the insulin-producing cells, the islets, the beta cells within the islets of Langerhans. These produce insulin. As a result, my body, and that of type 1 diabetics, is unable to produce insulin. So to treat this, there are really two primary interventions. First is to monitor blood sugar, and that's done through either a blood glucose monitor, so pricking the finger several times per day and evaluating what your blood sugar is, or wearing a continuous glucose monitor. And continuous glucose monitor, depending upon the model, will every minute, every couple minutes, provide a reading from the interstitial fluid of the patient's glucose. That's paired with insulin replacement therapy. And insulin replacement really comes kind of in two ways. Insulin injections, either with a needle or with a pen, or insulin pump therapy. And modern insulin pumps, they've been around for quite a few years, since I'd say probably the mid-'80s, more widely adopted in the mid-'90s, have a catheter that goes into the body and they continuously provide insulin, but they require constant input and they require monitoring by the patient. They're paired with, they can be paired with the continuous glucose monitors and what are called hybrid closed loop systems. These kind of came in the market about 9 or so years ago, and they essentially close that loop wherein the continuous glucose monitor talks to the pump and provides feedback. The pump then adjusts how much insulin is given based upon the blood sugar reading. Now, the uptake of these two different technologies has been pretty vastly different. They've been around for decades, for a very long time. And what you can see here, on the left are youths and on the right are adults, is that this continuous glucose monitors around 2015, 2017 had a dramatic uptake in use as compared to insulin pumps. You'd think that something that removes the need to give yourself several injections per day, right, would be widely adopted, that many adults and children would use it. But it turns out fewer than 30% of type 1 diabetic adults actually use insulin pumps. Why is this? Whereas the vast majority use continuous glucose monitors. You'll notice a difference in youths that the uptake has been trending upwards. But pumps have very limited clinical adoption. CGMs have widespread clinical adoption. It's different in youths. We'll discuss a little bit perhaps why if we have the time to do so. And for me, this adoption problem is personal. I didn't start using an insulin pump until 2013. I've been a diabetic since 1988. These pumps were available mid to late 90s. Why did it take me 15 years to come on board and use an insulin pump? I'll talk about that in the context of perhaps why CGMs and pumps are adopted differently, and why this perhaps occurred in that 2015 to 2017 area. You see that dramatic uptake in CGM use. Well, around 2015, insurance, Medicare, and Medicaid coverage and reimbursement was widely adopted for continuous glucose monitors. You can get pretty good coverage for medical devices or pumps with medical device coverage, but they're very expensive. There's perceived and there's actual benefit, and there's perceived and there's actual complexity differences between CGMs and pumps. Around that 2015-2017 period of time, CGMs went from needing to be replaced every 48 to 72 hours to needing to be replaced once a week. They went from alarming you several times a day, requiring calibration, to no longer requiring calibration at all for some models. They became significantly easier. They became integrated with smartphones, so you could get alerts not just from your device, but on your phone, and other caretakers could get alerts as well. So there's significant actual benefit. There's significant perceived benefit as well. Insulin pumps are difficult. They require action from the user, and they have a significant risk profile that is different than an insulin, or that is different than a CGM. A CGM provides information that's actionable. It takes away all those finger pricks. It really can be a lifesaver. An alarm in the middle of night can wake me up to a low blood sugar that I would have otherwise been ignorant of. These are real and actual benefits, and though monitoring insulin over time is complex, there's not the risk of error that you have with an insulin pump. There's a perceived and actual physical burden in that pumps can get knocked out. They're in the way. One of the reasons that I was late to adopt pumps was for these physical burden reasons. Oops. And then finally, there's a lack of familiarity amongst healthcare providers and an ability to support the adoption of use. And healthcare providers were very reluctant to use pumps, and to this day are somewhat still reluctant to use pumps because of the risk of error and the dramatic life-altering effects that can have with low blood sugars. Importantly, these three in the center here all come down to the patient perspective and the willingness of adoption. You're competing in a very crowded field with type 1 diabetic therapies. Whatever therapy is developed, it has to compete for my attention, it has to compete for my time, it has to compete for my money. I have hundreds of decisions to make every day regarding my diabetic therapy. I have limited time. I have dozens of appointments every year going to an ophthalmologist, primary care physician, endocrinologist, so on and so forth. Does your therapy demand more of my time? And finally, pump therapy and my CGMs alone cost me several $1,000s per year. So whatever therapy you're going to develop is competing for those dollars that I have and many other patients don't necessarily do. So when it comes to developing therapies for retinal disease, consider that they're always downstream of type 1 diabetes management. All right, how are they competing for priority in a type 1 diabetic's life? Diabetic retinal disease often occurs a decade or more after the development of type 1 diabetes. So patients have well-ingrained patterns of care and willingness to adopt therapies. How will your therapy integrate into what is already being done by that patient? Type 1 diabetes, as mentioned, has many long-term complications. And while vision loss is high up there, it's not the only one. And so it may not be the highest priority for that patient at that given time. And so as you're developing a therapy, ask yourself many of these questions. Will type 1 diabetes patients prioritize these therapies? How will it fit into the complex health management of type 1 diabetes? What are the burdens of adoption? And more importantly, what are the risks? Will your therapy complement what type 1 diabetics are already doing, or will it demand more of them, right? And if it demands more of me, what am I giving up? What money am I not spending in care otherwise? What attention am I not giving other parts of my life? And then finally, can patients afford this? So please think proactively throughout the intervention life cycle as to how you can engage patients in the discovery phase, in the clinical phase, in the marketing and approval and post-marketing. And while this may seem a request tailored to industry, to the developers of devices and drugs, as an academic researcher, you're part of this discovery process. If you're trying to develop a therapy, ask patients or try to find patients and ask if this seems like a reasonable concern that you're dealing with, and is this intervention that you're developing a reasonable thing to do? Will it provide a need that they identify? As a clinician, you're involved in post-marketing. You're involved in engaging in activities like this conference to share your clinical experiences and how they can be better managed. With that, it is time to move on. So I thank you. I apologize. This was a bit rushed, but if you have any sort of questions, feel free to send me your comments and ask me your questions at this email address, or feel free to send me a message or give me a call at this phone number. Thank you very much, and I will turn it back over to Jenny for the panel.
Thank you so much, Chris, and I'll invite all of our speakers back up here for the panel. Chris, while we're waiting for the other speakers, I'm going to go to you, because that was, a beautifully articulate kind of discussion of what are the key sort of drivers of whether patients use technology or not. And I guess I'll ask you, so from your standpoint, as we think about developing new severity scales, as we think about developing new endpoints, how do we engage patients in that conversation beyond what we're doing already?
I think there are not well-defined ways to engage patients, and that's one of the biggest problems. Activities like how the Mary Tyler Moore Vision Initiative has engaged the lay community could be illustrative of ways that other organizations can as well. They've created a lay advisory committee. They've actively sought out feedback from patients, and one of the biggest risks with that is that you get biased feedback and that you get limited feedback. We have a lot of feedback from, say, type 1 diabetics, but what the perspective of type 2 diabetics is wanting, for sure. But developing those panels is a great first place to start. I think the initiatives that CCOI is undertaking, trying to collect and aggregate as many perspectives as possible and glean information from that is extraordinarily valuable. And with the onset of AI, going through all of those different perspectives and coming up with consensus considerations is going to be key. But it's not easy. If it were easy, it would probably be more widely adopted. And patient perspectives and assessing patient perspectives is a very soft science, right? It's difficult to have measures and metrics that are meaningful and actionable.
Fair enough. And I will say, for those of you in the room who are representatives of organizations that connect with patients directly, I think we're very interested in building bridges and in helping each other to amplify our messages and our resources across communities. So please reach out to us, reach out to Chris. We're happy to engage in those conversations. I'll move on to the question about moving new endpoints forward. We've identified this as a key barrier in all of our conversations with industry, with academics, with people in these spaces who recognize that our current approvable endpoints are mostly relevant to late-stage disease and are getting harder and harder to meet in an era of better education, better systemic control, reasonably good therapies. These are all good things, but what it means is we need thousands of patients and years-long studies in order to get to studies that can address these in statistically significant ways. So Lloyd and Fabio, I'll ask the two of you, Fabio first from the industry perspective and Lloyd from the sort of academic scientific perspective. As we think about developing new endpoints, you both talked about retinal nonperfusion as an example of something that is at least partway perhaps down the path of that roadmap that Lloyd so beautifully presented. Fabio, I'll ask you first. From an industry perspective, how do you think about which endpoints you're engaging in the clinical studies that you're planning, and how do you develop, what are the thresholds for a new endpoint to meet your interest and willingness to participate in these studies?
Yeah, so first, Jenny, thank you so much for that. I think that's a really key question. First, I think in what happens in the latest years, we have seen a massive increase in knowledge so that we know better about what is the disease progression ongoing. So definitely what is important is first to locate those variables that are at the on the critical path for disease progression, and that can be influenced. And I think now we have a much better idea of what is, especially in diabetic retinal diseases. So from an industry perspective, the investment and the planning of analyzing those variables is really very important to have also an early assessment, because what we want to do, what is important from an industry, from a drug development perspective, I wouldn't say industry, from a drug development perspective, is to have an early assessment of if the treatment is actually effective, how effective it is, and on which patient, on which stage of the disease. So in that respect, the first point is to locate the variable. The second is how do we measure it? And I think the artificial intelligence here has opened up a big spot, because it is not very desirable to observe patients that are going worse. honestly, also from an industry and drug development perspective, this is not what we want to do. We want to save patients. We don't want to observe patients going worse. So in this respect, most likely going forward, and there is opening here with regulators, we may have a frank discussion on what makes sense to be done, to have an integrated artificial postdoc or, let's say, retrospective postdoc assessment of variables in order to make sure that we are highlighting what makes sense. But I don't know, Lloyd, what do you think?
With regard to the markers and the early progression of endpoints and early things to look at, completely agree. I mean, it's absolutely necessary, and I think it gives us, sort of a go, no-go type of thing for further development. I think where some of the problem rises here is what's been sort of set as a ability to have a true FDA registrable or any registrable endpoint. And the problem there is it's based on visual function, which of course makes sense. And in the early days, it made sense because that was it. But now there are things that we know may lead to the visual function decline, but it's going to take too long to do it, too many patients to show it. I mean, it was this way originally for PDR with the FDA. The FDA didn't accept PDR as an endpoint. It had to be two-step progression or three-line loss on vision. And the argument was finally that, well, if you get PDR, you have to have laser, and laser is a bad intervention and so forth, because you couldn't do the study to look at preventing PDR with vision because, 94% of the vision, severe vision loss from PDR is prevented by PRP, and you can't withhold the PRP. So we need to be able to get that concept where we can have an endpoint that's related to visual loss, even without waiting there, because now we can prevent severe visual loss, if that's going to be an endpoint, many, many ways and clearly. So.
And
Actually,
Malvina,
thank you. Can I just ask a question to Lloyd?
Yes, before you do that, I'm going to actually ask Malvina for her perspective from the FDA standpoint, for her years of experience. I'll have you guys do concise answers because we're running up against time, and have the audience also think about what questions do you want to pose to the panel.
So just real quickly, just to comment on the two answers, I would like to challenge everybody to think about consensus endpoints not being specific to drugs, but think of it as trying to come up as a community with consensus endpoint that makes sense for patients, irrespective of what kind of medical products. There's a lot of devices and a lot of combination products in the pipeline as well. And while the safety profiles will be different depending on the therapy, the efficacy should be consistent across all different modalities. Now, the last point, for the sake of being concise, I want to make is I want to make sure that everybody understands that the only way to move forward is to have everybody at the same table. It's not we're going to go to FDA and we're going to ask a question. Let's change the paradigm. That's exactly what CCOI is doing. FDA is a member of CCOI, as our other regulatory bodies are starting to join as well. Let's have the workshops that we have been talking about and the DRD Accelerator will put together.
Love it. Fabio, please ask your question.
Just wanted to say that, what do you think that, so first, totally agree with you, Malvina. This is CCOI actually is a great integration of all those. What we have seen, especially for retinal non-perfusion, but also for other endpoints, definitely technology progression has made a huge kind of step forward. How do you think that this can play in for the other endpoints too?
Well, I think it's made a huge step forward. It's also made it difficult because before we can fully analyze one technology, there may be a better technology coming, and then we have to put it together. So I think this is wonderful. The progression, you need to look at it, take whichever ones still look good and move them forward, but not necessarily wait for ones behind if you start to get the association, because all of this is changing, and 12 months from now, there'll be a whole new series of OCTs out and so forth that are going to change things. And so I think it's critically important, but to take the best you can and move it forward in the type of studies that MTM, for example, is trying to look at.
We have time for some questions from the audience. George.
Hi, this is George King from Boston. So I think Fabio or Dr. Kai,
Fabio's here.
Fabio's here.
Kai is with us in spirit.
Wasn't sure. I was just reading. I just want to make a comment and a quick question. And the comment, I think, is I totally agree. Negative studies are very important. You can learn a lot from. But I assume the hope is you get a positive study, right? That's I presume that's what the company wants and everybody wants. So the comment is that, you know, we're going to talk about human repository in a few minutes. You know, many of you have been saying this for at least 20 years now. I know Lloyd and Jenny's hearing this, is that the 90% of the animal studies are not translatable to complication for diabetes. So, and one major reason, it's not because the animal models are terrible, but is used not correctly. So for your study, where you showed STZ model, almost 90% of animal model publications are preventions. And many of you may not know that after 3 months of diabetes in rodents, the changes in the eye are not reversible. So but you're doing these clinical studies, it's always intervention. You don't start at year 1, right? So I'm just curious, for your study, where it was a negative study, was the animal model, STZ, 3 months after the induction of diabetes? Because again, 90% of publication, even today, after 20 years, I might keep talking about this, people don't do that.
Thank you for the question. It's actually very, very interesting. So obviously, we have presented, we have published just a paper together with Elia Du and the Johns Hopkins group that took care of the development, of the preclinical development of runcasikvat. So we did both. I think you're absolutely right. We did both in prevention, for example, in the oxygen-induced retinopathy, both in prevention and treatment, which is not a diabetic retinopathy model. We have what we have, actually more glaucoma, right, or ROP. But we did have more ROP, and then we did a kind of more glaucoma model, which is applying pressure on the anterior chamber to get, kind of nerve fiber damage, and as a pre-treatment and post-treatment. And we also have seen good data. It's more glaucoma model. You're right. There is not much. This is also why, as Boehringer now kind of stepping over on this my real toes, we have had an agreement with Michigan in order to start a collaboration. We have already started on human tissues. I think this is already a big step ahead. But the translatability of results from preclinical is definitely an issue. This is why most likely we will move into phase 1b data. Now here the nomenclature is tricky, looking at biomarkers in patients. And this is also why retinal nonperfusion, as well as the effect on drills and drolls and others, may be more important. I was not too long, sorry.
Perfect. And we're just getting over time. So Quentin, I'm going to give you last question, 30 seconds or less, and answer from the panel, 30 seconds or less, so we can get people to the break.
Great, thanks. Quentin Davis from LKC. And in thinking about developing a new endpoint that can be validated, Lloyd, you presented data showing that, you got maybe a factor of 1.7 or 2-fold increase in relative risk from looking at the retinal nonperfusion. Do we have an idea of what would be accepted by regulators of if you're trying to make a relative risk statement saying, if you had this endpoint, you were X% more likely to have disease? What X would need to be to make it a useful endpoint?
Malvina?
So let's take the guessing work out of the equation. Let's change the paradigm of how we do endpoints. Instead of saying, would the regulators accept it, why don't we get the regulators at the table with us and jointly come up with a gold standard consensus endpoints that are not something that we all need to guess about, that are published and well understood by everybody in this room and around the globe.
All right. Chris?
Well, I just have one comment about that, because I do work with FDA in the medical device world, and there are many times where we've had pre-submissions to FDA, we've gotten their feedback, we've agreed to study, clinical study and analytical study endpoints, and then in review, they come back and say, the reviewers might be different, and they come back and have different requests and state that previous guidance, is not necessarily binding, and they're not necessarily held to that. So I think there's quite a bit of apprehension in industry about what additional things may be asked during review that are not brought up beforehand.
So let me just explain that there's a difference between communicating on a sponsor to sponsor versus having a gold standard consensus that's published and that's perhaps recognized in standards as the gold standard that FDA will uphold.
Okay, Lloyd, last comment.
Of course, that's true, but you also, the question is still valid because in your meetings, in your develop to do that, you have to ask that question. And that's what the regulatory agencies do anyway when they do publish the few things that they're willing to stand behind. One's going to still have to answer the question, what is enough to be considered important by group consensus to set that as the standard?
Perfect. Thank you all.